Welcome to lecture 3. We're going to talk about data management, typical structure for data sets, and then do a quick review of descriptive statistics in R. And most of this we will probably start to do when we dive into exercise 1. Okay, so here's just a quick outline. We're going to talk about data set structure, data management and cleaning, our descriptive table or our table 1 that we typically would include in a peer-reviewed publication, reporting our results of any studies that we conduct, and then a review of assessing normality and outliers and how to do that in R. So data sets are typically rectangles where each row corresponds to our unit of observation. And for most of us in our research, a unit of observation would be a person. In other studies, like animal studies, they might be a rat or a Drosophila fly. But for the purposes of this class and for most of us, our research will be conducted in samples of people. So each row is a unit of observation, but each column corresponds to then a variable. That could be someone's age, it could be their height, their weight, and other characteristics that we are interested in collecting on those units of observation. A lot of us will probably collect data and enter it into an Excel spreadsheet. And so if that is the case, then the first row is typically where we would put our variable name. So we might have an identifier for each person in our sample, and then we would have a variable name for all of the different variables that we measure and include in our data set. Data sets can be long, and that just means there are multiple rows per person, or they can be wide. And so that just means we might measure a variable multiple times, and we have each of those measures in an individual column. And I'll give a visual of that in the next few slides. When we create a data set, it's good practice to also create a data dictionary that defines the variables in your data set. So that way, when you go back to your data set, or if anybody is interested in using your data, they can look at the data dictionary and have an understanding of what variables are included in your data set and how those variables are formatted. It just gives the user an idea of like a summary of your data set, and it makes it a lot easier to then move forward and utilize that data set. So if you are using a data set that you did not create, it's always good practice to check its structure and to review its data dictionary if available. And a lot of the data sets that we will be working with are publicly available, and they will have data dictionaries associated with them. So it's always good practice to sort of review that and have a good understanding of that data set before you then move on to do your descriptive statistics and analyses. Okay, so I mentioned that data sets can be long or they can be wide. So on the left-hand side, that's an example of a long data set. So in this very, data set with just a small number of variables, we have individuals, and then at different time points, we measure their weight. So in that first column, we just have an ID for each of the individuals in our sample. And then we have this time variable corresponding to the time point at which the weight was measured. It's always good to have a variable like this in any study that we do repeated measures, just to sort of anchor that time point with the measurement. So sometimes it might also be a date. So the date that we took that measurement for weight. And the reason why we do this is, let's say we didn't have that time column and we just had the two columns, ID and weight. And you can see the second individual in this sample is missing their weight value for time point 4. If we didn't have that time variable, we wouldn't know, which weight measurement was missing. So, you can see that we've measured weight 4 times in each of these individuals, but for individual 1002, they would just have 3 rows, and we wouldn't know, at which time point were we unable to measure their weight. So it's always good to have this like anchor variable corresponding to the measurement. So you can see here, there are multiple rows for each of those participants in our sample. On the other hand, we can have a wide data set. So that's just the data set that's depicted at the top right-hand side there. And you can see now we only have 1 row per participant. So we have, now we don't have that time variable, but we have 4 weight variables corresponding to each time we measured that participant's weight. So you can see participant 1002 didn't have their weight measured at time point 4. And so you can see here in that Y data set that it's also missing from that column. This is an example of a data dictionary, in this case for the long data set. So here in the row, we just have, in the top row, we just have a description of each column. So in this first column, we have the variable name and then a description of that variable. So we have ID, we have time, and we have weight. And then here's just a quick note, it's good practice when we have numeric variables or even categorical variables, it's good to indicate the unit of measure. So these values, if we look at that and we know that it's weight, it's more than likely it's in pounds, but it's possible if we're studying an obese population that these could be in kilograms. So it is always good to indicate your unit of measure. If these were categorical, it would also be good to write out the categories in your data dictionary. So for example, if the first category corresponded to individuals of a certain weight range, we would write that out, and then the second category and third category and so on. And we'll see more examples of this as we move on through the semester. And this would just be the data dictionary for that wide data set. So here again, we have the ID variable and a description of that. And then here we have the weight 1, 2, 3, and 4, and we can put them all together in this case. And then we've just described it here, the weight in pounds at each time point. So what do we mean by data management? Because that really comprises of a lot of our time when, even when we receive data sets that are publicly available, for example, in Haynes data or other national survey data, they're still pretty raw and messy, even though there has been a little bit of cleaning, on the data collector's side, we still are receiving data sets that might not necessarily be ready for analysis. So it's often very good to, whenever we start to work with a data set, to check it out and to look at the structure and the summary and get a summary of your data. And you can do that using these functions in R here. Another thing that we might need to do when we receive a data set is, for example, to modify variables. So let's say we have a continuous variable in our data set for age, but we need to categorize age, before we run our analysis. So we might end up categorizing numeric variables. And another thing that we might do is we might need to reassign values to variables that are in our data set. So these are all considered, data management and preparing our data for analysis. We might also need to create variables. So let's say we have a data set that includes height and weight, but we're interested in using BMI in our analysis. We would then create BMI from the height and weight data. You might also be interested in only a subgroup of our sample, so we would subset the data. So for example, we might just restrict our sample to women. And then another thing that we often do prior to analysis is we need to clean up our data. So we need to assess missing data, any outliers, and decide what to do in those cases. Do we exclude the individual? Do we exclude outliers? And then also looking at values that might be typos or data entry errors. So for example, the cohort that I work with, through one study, we identified these extreme values of weight and height. And then it turned out when we went back to the original survey, we saw that it was most likely a data entry error into our database. And so we were able to correct those and keep those individuals in our study. So all of that, it makes up this data cleaning process. And that's very important and will take a lot of your time, but it's very important to have a nice, clean data set that that is ready for analysis. So I showed this table in the last lecture, and we're actually going to be creating a table 1 in our exercise today. And table 1 just describes your study sample. So for discrete variables, we would present frequencies and percentages. And then for continuous variables, we might present the mean and standard deviation, or in the case of non-normally distributed data, we might choose instead to display the median and the range or the interquartile range. Table 1 is often stratified by a specific characteristic of interest. So for example, we might want to compare men and women, or we might stratify by the outcome of interest. So if we're interested in studying a particular disease, we might present our data and include descriptive characteristics, and then stratify those by no disease versus yes disease. So in this table, we see that the authors are presenting demographic characteristics, so age and sex, and then clinical characteristics, so obstruction and perforation. And these are all in categories. So remember, we talked about categorizing age, and it's sometimes a lot easier to present data this way. And then they've just stratified by whether an individual was alive or dead at the end of their study. And then I asked you in lecture 2 to kind of ignore these p-values, and I'll ask you to do that again, but we will talk about hypothesis testing and specific hypothesis tests later on, but I will just briefly mention that in this particular case, these p-values correspond to a chi-square test because we have categorical values here. Okay, and then just a quick review on normality and outliers. So we do want to check for normality in our data, and then also for outliers and decide what to do with that. So there are a number of ways that we can check for normality. We can plot histograms, density plots, which really just look like smoothed histograms, or we could also plot stem and leaf plots. So that looks like a sideways histogram, and instead of filled in bars, we just have the actual data points that make up the bars. Another way we can check for normality is using QQ plots, which we discussed in lecture 2. And then there are a number of statistical tests that we can use to check for normality as well. So the first test, the KS test, it's really actually just a general purpose test. So in R, when we implement this test, we do have to specify the distribution for a one sample test. So in our case, we would include our variable, and then we would specify that we're testing against a normal distribution. This test can also be used to look at two different samples, in which case we wouldn't have to specify a distribution. In that case, it's just asking R to assess whether our two samples are distributed in the same way. Another test is the Shapiro-Wilk test. And then there's the Anderson-Darling test. And I know I didn't mention that in lecture 2, but I put that there because we do have example code in that R example code file that I posted to Cougar courses. So that is another test that you can use to assess normality. And then to check for outliers, the most common visual way to do that is to plot box plots. And that can be done using the box plot function. This is sort of in just regular base R, but there is also this box plot with a B, capital B, and remember R is case sensitive, and that function is available in the CAR package. And the nice thing with this second box plot function is that it does label outlier points. So whereas this one, it will show outliers as circles, but it won't actually identify which observations those correspond to. So this second capital B box plot function might be a little bit more useful in that case. And then a statistical test that we discussed that we can use to assess outliers is the Grubbs test, and that also is available in R. So that's all I had. It was a very quick lecture 3. Most of our time on Tuesday will be spent working on probably the first 3 questions in the exercise. So if you get a chance, take a look at that exercise. See if you can import the physiology data. If you're not able to, that's okay. We're going to work through that on Tuesday. And then take a look at the R sample code as well. That will give you a lot of the tools that you'll need to start getting at questions 1 through 3 on that first exercise.